In this lecture, we'll study about gluconeogenesis, the synthesis of glucose from non-carbohydrate compounds is known as gluconeogenesis, and the major substrates or precursors for gluconeogenesis are lactate, pyruvate, glucogenic amino acids, propionate, and glycerol. Gluconeogenesis occurs mainly in cytosol, that is outside mitochondria, although some precursors are produced in mitochondria. Gluconeogenesis mostly takes place in liver and to some extent in kidney matrix. Next we shall study about importance of gluconeogenesis. Brain and central nervous system, erythrocytes, testes and kidney medulla are dependent on glucose for continuous supply of energy. Human brain alone requires about 120 gram of glucose per day out of about 160 gram needed by the entire body. Glucose is the only source that supplies energy to the skeletal muscles under anaerobic condition. Gluconeogenesis is essential for the survival of humans and other animals. Certain metabolites produced in the tissues accumulate in the blood, example lactate, glycerol, propionate, etc. Gluconeogenesis effectively clears them from the blood. Next is reaction of gluconeogenesis. Gluconeogenesis closely resembles the reversed pathway of glycolysis, although it is not the complete reversal of glycolysis. Essentially, three reactions of glycolysis are irreversible, and the three reactions are catalyzed by the enzymes hexokinase, phosphofructokinase, and pyruvate kinase. And these three stages are bypassed by alternate enzymes specific to gluconeogenesis are discussed. In that first is conversion of pyruvate to phosphoenolpyruvate. Second is conversion of fructose 1,6-biphosphate to fructose 6-phosphate. And last is conversion of glucose 6-phosphate to glucose. Now, the first step is conversion of pyruvate to phosphophenolpyruvate. This takes place in two steps. Pyruvate carboxylase is a biotin-dependent mitochondrial enzyme and converts pyruvate to oxaloacetate in presence of ATP and carbon dioxide. This enzyme regulates gluconeogenesis and requires acetyl coenzyme A for its activity. Oxaloacetate is synthesized in mitochondrial matrix and it has to be transported to the cytosol to be used in gluconeogenesis. Due to membrane impermeability, oxaloacetate cannot diffuse out of mitochondria. So, it is first converted to malate and then transported to the cytosol. And then it is regenerated with the help of enzyme malate dehydrogenase, an enzyme present in both mitochondria as well as cytosol. And in cytosol, the phosphophenolpyruvate carboxykinase converts the oxaloacetate to phosphophenolpyruvate. Next step is conversion of fructose 1,6-biphosphate to fructose 6-phosphate. Phosphophenolpyruvate undergoes the reversal of glycolysis until fructose 1,6-biphosphate is produced. The enzyme fructose 1,6-biphosphatase convert fructose 1,6-biphosphate to fructose 6-phosphate. This enzyme is also called as regulatory in gluconeogenesis. Third step is conversion of glucose 6-phosphate to glucose. Glucose 6-phosphatase catalyzes the conversion of glucose 6-phosphate to glucose. The presence or absence of this enzyme in a tissue determines whether the tissue is capable of contributing glucose to the blood or not. It is mostly present in liver and kidney, but absent in muscle, brain, and adipose tissue. Next, we shall see about gluconeogenesis from amino acids. The carbon skeleton of glucogenic amino acid results in the formation of pyruvate or the intermediate of citric acid cycle, which ultimately result in the synthesis of glucose. We shall study about regulation of gluconeogenesis. The hormone glucagon and the availability of substrate mainly regulate gluconeogenesis. Glucagon reduces the concentration of fructose to 6-biphosphate. This compound allosterically inhibits phosphofructokinase and activates fructose 1,6-biphosphate. Both favor increased gluconeogenesis. And among the various available substrate, glucogenic amino acids have stimulating influence on gluconeogenesis. Acetyl coenzyme A also promotes gluconeogenesis. During starvation, due to excessive lipolysis in adipose tissue, acetyl coenzyme accumulates in the liver, and acetyl coenzyme allosterically activates pyruvate carboxylase, resulting in enhanced glucose production. But alcohol inhibits gluconeogenesis. Ethanol oxidation in the liver to acetaldehyde by enzyme alcohol dehydrogenase utilizes NAD+, and the excess NADH produced in the liver interferes with gluconeogenesis. So from this reaction we can say that ethanol is treated with NAD+ and produces acetaldehyde, NADH and H+. So these NADH and H+ is further treated with pyruvate and gets converted into lactate and NAD+. Again, NADH and H+ is further treated with oxaloacetate and gets converted into malate. So over here we can say that the precursor or substrate which are required for gluconeogenesis, that is pyruvate and oxaloacetate, are used up when it is treated with ethanol. Hence, we can say that alcohol inhibits gluconeogenesis.